Okay, yeah, thanks for having me. I think that was a great lead up to what I'm about to talk about now. So, I'm really interested in autonomy. I think it's probably the fundamental principle that we really mean when we talk about intelligence or animals having an adaptive behavior. So, the way I would define it is autonomy is kind of the capacity for an organism or an agent to motivate behavior by having internal states, right? So, if you think about like an automaton would purely be extrinsically motivated, whereas animals seem to have this ability to have many different intrinsic goals. Some of them are physiological, like food rewards. Other of them are more abstract, like curiosity or play. And so you get this kind of spectrum where you have intrinsic drives, like I mentioned here, but then you also have these extrinsic drives where the extrinsic part is defined in terms of being externally conditioned. So they can appeal to primary reward mechanisms that Lillian mentioned, but the key distinction is that they're extrinsically conditioned. But really, this actually kind of defines a spectrum of different behaviors, right? And so if you take out a trajectory of like any kind of intelligent behavior, you're really tracing out different amounts of intrinsic or extrinsic rewards or motivating signals. Like on one end of the spectrum is purely intrinsic. You imagine you're dreaming, where like your state in your dream has really no tie to the physical world. It doesn't need to be commensurate or reconcile with anything you've seen before. Or you could be like a robot or like a cockroach where you're just purely driven by an extrinsic signal that you repeat and it's some deterministic input-output function. And so the easiest way to kind of capture what I mean by autonomy is this video of this baby just playing. So you might have seen this before. And when we look at an animal do a behavior like this, why is it interesting, right? This is clearly something we would call intelligence, but doesn't fit into a goal-driven behavior, right? So if I asked someone, say, like, what is the goal of this? Can't say, like, oh, maybe it's supposed to touch everything in the room with equal probability. That's not really a goal, right? And if we even define the goal like that, there's no really clear success rate. Like, what does success mean for this baby, right? Is it supposed to see or smell or lick all of the floor or something, right? It doesn't have a clear supervised signal, so it's difficult to computationally formalize. And in both neuroscience and machine learning, we usually are in the setting where goals are easily identifiable, right? We specify a task, and once we specify the task, we kind of ascribe intelligence to what's going on relative to that extrinsic goal. So in reinforcement learning, what this looks like, right, this classic picture is that you get reward from the environment. But this baby, right, there's no one telling it what toys to play with or where to go in the environment next. So if we remove this stream, right, this is kind of what Lillian talked about, we don't have an extrinsic reward from the environment. A lot of these intelligent behaviors, the alternative is that the agent generates its own reward, right? So the reward function is a function of the parameters of the agent itself, rather than being a function of the extrinsic state of the world. And so the goal of our work is try to come up with algorithms for this intrinsic function, this intrinsic learning signal that capture the richness of animal autonomy, like play, exploration, things like that. And so to do that, we need a neuroscience data set because we want to be able to validate these algorithms on real behavioral data and neural data. And so what we use is zebrafish data. So the reason why we do this is because it turns out zebrafish, you actually don't usually train them to do anything because they're fish. And so almost all of their behavior is autonomous. And so I've highlighted where the fish is right now. This is the fish swimming in virtual reality. So there's electrodes that are recording motor neurons in this tail, and those motor neuron activity based on the actual like profile of that wave determines turning and the speed of the ground plane moving against it. And if you notice here, there's this passive sort of flow. This is because it kind of simulates the optic flow of being pushed against the current. And so zebrafish have this homeostatic drive to oppress that. So they just like to swim. And so the behavior that I'm going to be talking about is something called futility-induced passivity. So if you've heard of learned helplessness, this is very similar. Because you're in virtual reality, what you have is you can mess with the sensory motor loop. So in the closed loop, what you have is motor commands that actuate the ground plane and produce sensory feedback in the form of optic flow. And now, because it's in virtual reality, you can break that sensory motor loop. You can set that control gain to 0. And when it sends motor commands, it gets no sensory feedback. And so what the animal actually does is after repeated tries of doing that, it gives up, okay? And after a little while, it'll try again and then give up and so on. And so why is this behavior interesting? Like why would it give up? I mean, the simplest answer is just that it runs out of energy, right? It like doesn't want to just swim forever. But so this data is from Genelia, and they did a lot of control experiments to actually show it's not due to some metabolic cost or energy constraint or some misunderstanding of how the experiment is set up because it's in virtual reality. It really is in some internal state where an animal doesn't like to be in a position where it doesn't have affordances in the environment, doesn't have the opportunity to control. It's not driven by just like wanting to save its energy for another day. And we can also see this in the neural data. So if we look at larval zebrafish, its brain is transparent, so we can record over 200,000 units here between neurons and astrocytes. And what I'm showing you, this is an open loop, again, from Genelia Lab. well, you see this really nice separation of computations between noradrenergic neurons and GABA and radial astrocytes. So what's happening is while the fish is silent, it's in open loop. What happens is noradrenergic neurons will fire when there's failed swim attempts. So this is whole brain activity. And then radial astrocytes accumulate that calcium and send it downstream to motor neurons that end up getting inhibited by the GABAergic neurons. And so another reason this data set is interesting because these astrocytes play a really critical role in mediating this behavior. Now, if these neurons over here are computing this blue line here, right? They're only firing when the fish is active in open loop. That means they're computing some type of sensory motor prediction error, right? Similar to like certain neurons in other areas of the brain that would be computing a reward prediction error, which means you have to have a model of reward. So in this case, the animal doesn't have a model of reward, but has a model of sensory motor dynamics, right? Because otherwise, there's no signal to compute an error with respect to, right? It has some model of how the dynamics of the environment should evolve under normal circumstances. Okay, so with that, we wanted to capture this in an embodied agent. So this is the virtual zebrafish design. It gets vision and proprioception. Those feed into a recurrent actor-critic kind of reinforcement learning agent. The most important detail here is just that we have this intrinsic drive module that takes in the features. So these are the observations these are the states of the zebrafish in observation space, and builds some intrinsic reward function. Now, again, because we know that we're in the space of having a world model, a sensory motor dynamics model, because of those neurons prediction error, we're in the space of intrinsic motivations that are model-based, that they're building models of the world, predictive models. And so our R here is going to be all based on world model as the fundamental substrate, and we're going to implement a bunch of different algorithms from reinforcement learning, most notably like random network distillation. There's also disagreement. And what we're going to show is that the algorithm that we propose actually allows us to go back in and color in the units of the RNN in our network with the exact kind of circuit motif that the scientists at Geneli identified. Okay, and then so the actual implementation of this is all in Mojoko. It's a physics simulator. There's some fluid dynamics. The important part here is that we also build a simulation of the experimental protocol in which the data was collected. That way we have a realistic comparison between neurons in our artificial unit, in our artificial zebrafish, and neurons in the real zebrafish when we do our comparisons. So before I talk about the algorithm we came up with, I just want to show you some results. So what I'm showing here is activity traces of the zebrafish over a single trial, that's the x-axis, throughout different points in learning. And these algorithms, some of them are like from Jürgen Schmidhuber's lab, some of them are from other kinds of approaches to curiosity. You can see over the course of training, they stabilize to a flat policy, right? Over the course of the episode, they stabilize to just a single decision-making strategy. Whereas what we're looking for, I guess I should just show this plot. This is a single trial of zebrafish. You get this barcode. So here, black is active states and gold is passive states. You see it mostly stays in passive, and it does an exploratory burst out of that state, out of that passive state to check if the world has changed, right? That's why you would go back to black. And then the world hasn't changed, so it goes back to gold again. And you get this barcode. So you can see this algorithm does that. I'll explain why in a second. But why do these existing methods fail? The most obvious reason is because when we've designed self-supervised reward functions in reinforcement learning, they kind of use the world model as the substrate literally. So your intrinsic reward is always a function of your prediction error, which means your prediction error is changing as the learner progresses in the environment. And so it's non-stationary and saturates to a fixed value if you completely learn on everything in the environment, which is a really unstable signal for a reinforcement learning agent. The other issue is that some of these actually reward aleatoric uncertainties. What I mean by that is if there's like some big entropy generated in the environment, some stimulus that's really random that you can't predict, some of these reward functions won't distinguish that and will say that's a stimulus you should perseverate on. But real animals don't perseverate on stimuli they can't control or can't predict. Okay, so to fix this, we kind of took inspiration from both the neural data, but kind of in the lens of, since we're in Portugal, there's this guy, Henry the Navigator, right? You've all heard of him. So he was kind of famous for a lot of things, but one of the things was that he was a big advocate for using what's called a Mariner's astrolabe. I think that's how you pronounce it. That measures basically the angle from your ship to the sun to see where your latitude coordinates are. But the other thing he was a big fan of was the compass. And so the thing that these two things have in common is that they're both one-dimensional signals that give you a very clean readout of where you are relative to some fixed point, right? So like a compass says, I'm pointing this direction relative to north. It's a very low-dimensional signal. That's really fundamental. Just you don't have to go north, but you can go in any direction relative to north. And so for us, the question of when you're exploring an environment, you want to discover a new policy, what is the north for behaviors? What is your prior? And so the way that we formalize this is by thinking of animals get this prior by developing an ecological niche, right? So they swim around in their environment in this case and build up a memory or some sensory motor bias towards the dynamics in that ecological niche. And then when they're in an unseen environment, the ecological niche provides a dynamics prior. So imagine that you have some prior here, that's the red guy, and then you have a new memory in this new environment, that's the blue guy. And in this new environment, what you're gonna do is you're gonna try to look for, or at least try to find states in the environment where the subspace of your dynamics agree between your memory and your past experience. So what that would look like is you picking actions that move you into the subspace. So that's like this first red trace, and then you can pick an action that more looks like the dynamics of your memory, and that moves you out of the subspace, and so on. So basically, you have these two memories that are guiding exploration in a structured way. Here, your memory of your ecological niche is like your north, where you can use that to basically structure which directions in your new space you explore relative to. And so the way that we actually formalize this is we have an active pre-training stage and online exploration stage. So in the pre-training stage, the fish just moves around in an ethological environment, so it can freely swim, it can go pursue food rewards, it can do all kinds of other things. There's currents that it has to oppose. And in the space, it builds up a prior about the world, right? So this is going to act as its sensory motor prior. So it learns a dynamics model in that ethological niche. And then online exploration phase, which is like the actual experimental protocol, what is here, it's actually head fixed, right? So it can't actually swim. It's trying to, but then it's going to build up a new memory of what the physics look like in that environment. And the residual between those, that's your orientation. That's your compass, right? How far away your epsilon is relative to your memory. And so because these are distributions, that compass is in KL, but it's really just a distance function. And what that loosely does, because it's either 0 or positive, is partitions the space into sort of two modes, niche seeking and niche avoidance. So in niche seeking mode, you're going to try to drive this epsilon to 0, where your memory and your model align. And then in niche avoidance mode, you can leave that subspace and drive your models apart. And so in order to avoid being in a fixed point and get some adaptive behavior, we want a way of kind of computing a derivative in a biologically plausible way. And so we just do that with an exponential filter. So we know the brain can implement something like this, individual neurons can. And so this, by comparing these two signals in time, you get sort of an adaptive, like a derivative over the pink guy. And so by tracking that, you can see how long you've been in a single mode. And then for the actual intrinsic motivation, what we do is we just compare those signals in time. And what you get is kind of like straddling the edge of chaos. So because there's a symmetric activation function on these guys, you want to be in the niche seeking mode, but not for too long. And then you'll want to leave to see what's going on outside the world, and you want to come back. So it's always relative, though, to this partition of trying to find that subspace where your memories agree and leaving and entering, like straddling that perimeter. And so what we want to do is now compare this to neural data. And so what I'm showing you here is this is sort of like our inter-animal alignment ceiling. So whenever you compare a neural network to brain data, you have to ask what kind of transform class you're going to use. For monkeys, right, we have to use linear regression because you can't predict a single neuron from a single neuron. You need to weight a population. For zebrafish, actually turns out if you take two individuals doing the same task, with perfect accuracy, you can find a neuron in the target that perfectly predicts the source, and vice versa, just with perfect Pearson's correlation. So it's a really strong metric because you don't get to weight neurons, right? You just have to pick a single other neuron or glial unit as your predictor of another neuron or glial unit. But it turns out you can do that with 100% accuracy. So for any neuron, you just find the most correlated one, and that's a perfect predictor of the other zebrafish neuron, which is pretty crazy. But it means it's more stringent, right? Because for the neural network, we don't get to weight all the units in the RNN to try to predict a single one. We just have to find a single unit in the RNN as well. But it turns out it's not actually too bad. So by actually constraining the behavior to match the real ethology of the zebrafish and actually getting active and passive transitions, that's enough to constrain the internal representations to model the neurons and glia on a single cell resolution. So we can go in for every glial cell and every neuron, we can find an RNN unit that perfectly predicts it. And now if we kind of plot this against the behavioral alignment, what we're seeing is sort of, we get pretty high on both. What this is trying to say is that if you can get basically a high model brain alignment, which is this guy over here, also model behavior alignment where you've actually kind of, the behavior of the zebrafish is kind of indistinguishable from the real behavior. It's kind of like a neuro AI Turing test almost. What we're after is like models of the brain that actually walk the walk. So like you build a model of the brain, prioritize behavior first, and that's enough to, if you can walk the walk of the real animal, it's enough to constrain the neural dynamics of the the system in this case. The last thing is we can also look at what happens to the circuit at the population level. So if we take those RNN neurons and just look at the PCs, we can see that both for active to passive and passive to active transitions, the PCs of our agent on the left directly reflect the computational motif of this neuroglial circuit. So where the first PC is like basically the computing the error signal, the noradrenergic neurons, and the second PC is like acting like the astrocyte's radial integrator. And so sort of the outlook here, the promise of kind of this approach to neuroscience using an embodiment is on one, you can propose and validate the circuit hypotheses from systems neuroscience in closed loop, right, at the macro scale. You can actually see if there's a behaviorally relevant circuit, if you embed that in a reinforcement learning agent, in this case, an intrinsic reinforcement learning agent, because the behavior is intrinsic, you can validate how that circuit is causally required for behavior. And the other thing is by forcing yourself as behavior as the primary readout, you're setting the bar high by having an algorithm that first matches what is the actual sensory motor capacity of the real animal. Let's match that first, and then let's see how the solution to that actually aligns with what we know about the brain. Is it very far, the algorithm that we came up with to solve this AI problem, or does it actually align with what the brain computes? And so with that, I'd like to thank our funding and my advisors, and we'll take any questions.
Reece, that was amazing. I had a quick question. Maybe it went a little bit too fast for me. So I understand that it's super important to have these dynamics that push you towards and away from your ecological prior. Two questions about that. I didn't understand what's exactly the process that drives that transition back and forth, and like, is there a normative reason for wanting to do that?
That's a really good question. So I'll answer the second one first. There's not, I think, a normative reason why you should straddle the most chaotic point, but I think it hints at the kind of edge of chaos kind of computational idea where you want to be at near a critical point, but not exactly at it, because there's the most information capacity at that point, like in a dynamical system. And so for that reason, we apply the same idea where it's not a normative model. We add that, it's really the symmetry in the activation function that it's an absolute value rather than like a ReLU or something that says after being in your niche for a while, you're also rewarded once your filter decays to go leave that. And that's a modeling choice because that's what we see in real animals. It's just exploring inside and outside of that niche. But it's not a normative thing that you need to have a symmetric activation like that.
Very impressive work. I really enjoyed. I'm a bit confused. So the way that you define intrinsic motivation and intrinsic reward is between going back and forth with this part of the environment that you already know, with the part of the environment that I don't know. And this resemble a bit to using these count-based and novelty-based intrinsic motivations, but with a very particular representation. And if I understood correctly in your benchmarks and baselines, those models didn't exist, like work of Mark Bellemare. How do you think those models would work? Do you think that would you get behavioral alignment but not brain alignment? Would you get both?
Yeah, so I think a count-based model. So a count-based model is basically like, I think in probabilistic terms akin to surprise, right? Like if you're counting novelty, it's very similar to just maintaining a distribution that you've seen over the states of the environment, right? That's what for continuous states, count-based is building a distribution of the environment. And so it's very similar to surprise. And so what would happen is if you just didn't have this prior, it'd be like your model's trying to learn on everything in your new environment. So eventually you would probably get these transitions, but it would decay again. Novelty would be zero because there's nothing novel in the new environment without a prior. The prior that adds a fixed structure where it's novel with respect to that. So if you had that, I'm sure you could design a novelty-based method that was similar, but just novelty alone doesn't have the right mechanism because like the other mechanisms, it only builds up the distribution over the environment you're currently seeing. And so eventually that novelty will flatline after a certain point.
Yeah, we can take
yeah, we can talk more offline.
Any other questions?
Very interesting. Have you looked at the temporal dynamics at all? Because what you would assume is that the natural statistics of change in the environment should actually guide exploration.
The temporal dynamics of the actual
when you go back and exploit.
So this is like over the course of a trial centered around when it actually switches. So the temporal dynamics of the neural data lines up, if that's what you mean.
No, I mean the frequency, basically. So if you have an environment where you expect change at a high pace,
like very volatile environments would mean that you should actually have a higher exploration frequency, as opposed to when it's a very stable environment, then it's actually less sensible to go back and explore.
Yeah, that's true. So we didn't look at that just because we don't have data for that specific case. The data we have only doesn't modulate the ground plane control gain in a stochastic way where it's like unpredictable or anything like that. But I think that's interesting. If there was a behavior we could compare against with that in real zebrafish, that would be interesting analysis.